DNA replication is actually quite an important topic for the IB student because it comes as a frequent essay question for section B in the paper 2. Now when you talk about DNA replication, students need to know the actual process, the enzymes that are involved, as well as at what point of the cell cycle does DNA replication take place. So usually at the start of a discussion on DNA replication, I emphasize that this process always takes place during the synthesis phase or the S phase of interphase. And furthermore, during the discussion on the process itself, I will repeatedly emphasize that replication always happens in a 5' to 3' direction. When discussing the process of DNA replication, it's usually helpful to provide the students with some kind of illustration or even a video is usually helpful for students to visualize the whole process. The 5 enzymes that you need to explain to the student are DNA helicase, RNA primase, DNA polymerase 3, DNA polymerase 1, and DNA ligase. So all the students will be required to know the names of these 5 enzymes and be able to discuss their separate functions. So the process of DNA replication, which happens during the synthesis phase of interphase, begins with DNA helicase unwinding and unzipping the DNA molecule by breaking the hydrogen bonds that hold the 2 strands together. This creates what is known as a replication fork, with 2 unpaired parent strands being found as templates. Onto these strands, the RNA primase enzyme produces short segments of RNA called primers so that the third enzyme, DNA polymerase 3, can bind and begin the actual process of replication. DNA polymerase 3 can only carry out replication in a 5 to 3 direction, which means that on one of the parent strands, the DNA polymerase 3 will be moving towards the replication fork, resulting in continuous replication, and we would refer to that strand as the leading strand. While on the other strand, in order to carry out replication in a 5 to 3 direction, the DNA polymerase 3 molecule will be forced to move away from the replication fork. As a result, it produces short fragments of DNA called Okazaki fragments, and this particular strand will be known as the lagging strand. DNA polymerase 3 carries out replication using free-floating nucleoside triphosphates and the principle of complementary base pairing. It's important to explain to the student the differences between the leading strand and the lagging strand, as well as the production of Okazaki fragments. However, prior to the completion of DNA replication itself, those short RNA primers that were initially laid down by RNA primase will be selectively removed and replaced with DNA nucleotides by DNA polymerase 1 before the process is rounded off and completed by DNA ligase, which connects the short Okazaki fragments together. This would complete the process of DNA replication. And I would close by emphasizing to the student that DNA replication is a semi-conservative process.